feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Loo School of Medicine Healthy Longevity webinar. Thank you so much for joining us. Professor Youssef Idaghdour is with us tonight, and we are discussing for me, an important topic, our genes, our book telling us who we are, or maybe not. I'm sure that this will trigger many questions, so please use the Q&A function and send us your questions. We are starting, as always, with a short presentation, and today it's from Ruslan Gumrov, a PhD candidate at NUS, and he is presenting its own research, applications of CRISPR screening and perturbation modeling techniques to aging research.
Thank you so much for the introduction. As mentioned, my name is Ruslan. I work with perturbation modeling and CRISPR screening in the context of aging research. At first, let me probably give you a brief introduction. What is the perturbation screening? When you have a cell, you have around 20,000 genes inside it. And well, the key question of drug discovery is, what are the genes we need to perturb, to interfere with, to get the beneficial phenotype? When the cell is diseased, we'd like to modify something in it to create a novel state of the cell that is resemble the healthy one. We are doing this by introducing perturbations to the cell, usually with the use of CRISPR, and then we can collect the readouts from the cells. We can get proteins, RNA expression, chromatin accessibility, and it's actually kind of cool. We can explore this space of perturbations and see whether we can find any meaningful groups in it. For example, here, you can see that after introducing a number of different perturbations, we can see clusters that are related to some specific cellular function. And when you're looking at this cluster of histone acetylation here on the top, for example, if your gene is laid down in it, you can think that, oh, probably this gene is related to this process. That's how it works. But what's even more cool about that is that when you're introducing, that you can potentially theoretically introduce double perturbations in the cell. And sometimes it's actually produced the phenotypes that are very different from single perturbations. For example, here you introduce HUS1, you can see this expression profile. You introduce other gene FDPS, you can see this one. But when you combine them all in a double perturbations, you can see this profile that's actually very different from the single gene post-perturbational profile. That's kind of cool because that's the way how you can find novel transcriptional states, novel states of the cell. And that's also how you can decipher, for example, the inner regulatory networks of the cell as well. So it's just the overall methodology is very exciting. But the thing is, it quickly adds up when you have more than whatever 1,000 perturbations and you'd like to do a screening of all possible pairs. When you have 10,000 perturbations, single perturbations, you can just screen them in the lab. It's okay, you can do this. But if you'd like to screen all possible pairs of them, you can get almost 50 million perturbation pairs, and that's just a lot of lab work. It's impossible to do that. And for triple perturbations, it's even more than that. So the thing is, we would like to find a way to generate these expressional profiles, post-perturbational expressional profiles, with the use of machine learning techniques. It's been a hot topic recently. Google, for example, released their model a week or 2 weeks ago. And the core idea of this model is to simulate this virtual cell kind of thing. You have your model, and when you tell it, oh, I'm going to perturb this gene, what the expression profile is going to look like, you have this expressional profile without an experiment, just from your computer. That's kind of cool. And that's also the way how you can find novel, interesting transcriptional states that might be beneficial. And that's what we are working on right now. For now, we are mostly benchmarking publicly available models. It turns out that they're actually not that great, unfortunately, for now. So we would like to create our own model after that and use this for the exploration of this perturbational space. And then we're also going to generate one of the data sets I mentioned before, specifically for aging-related perturbations, and see whether we'll find any novel, interesting interventions from it. For now, we're mostly on the benchmarking stage. We're trying to understand whether existing models and the whole concept of this CRISPR screen is actually meaningful. And this is one of the picks we made during this benchmark. We try to perturb the genes from the Krebs cycle, get the expression profiles from public available datasets, and see whether they're grouping to something meaningful. And yeah, it seems to be the case. We can see that we observe a lot of ATP-related pathways, glucose metabolism, oxidations, and so on. It means that, yeah, we're getting a meaningful expressional profile, and the space itself is very meaningful. That's exactly what we wanted to see here. And that's actually our, further plans on this. We'll take the models, select the best one out of them, and use it to help us to generate this novel data set of aging-related CRISPR perturbations. And then we'll try to find novel innovations with the use of it. I think that's the whole idea of the research. I wanted to say thanks to my lab, specifically to Jan Gruber and Brian Kennedy, and also for all of the lab members who support me during this research. Thank you so much, guys, and thank you for your time.
Thank you so much, really introducing CRISPR screen and perturbations to the aging field. We are staying with the genes. Our guest today is Professor Youssef Idaghdour, a geneticist and director of the Public Health Research Center at York University Abu Dhabi. He leads an international research program across regions like the UAE, West Africa, Morocco, and Mongolia, where he explores the genomics of complex traits, infections, and lifestyle transitions. His work has earned the York University Abu Dhabi Faculty Distinguished Service Award and the Times Higher Education 2023 Award for the Best Middle East and North Africa Research Projects. It's my honor, Joseph, to have you here. I really want to explore the genome and if the destiny are my genes, or maybe I can change the environment. Over to you.
Thank you. Thank you so much, Andrea. Can you hear me?
Perfectly.
Perfect. Yes, so the ultimate debate, nurture versus nature. So that's going to be the topic of my talk. So the big questions that my lab is interested in really revolve around these fundamental questions that has been, even the topic of like debates between philosophers and more recently scientists. And so the way we like to think about these questions is really to think about genetics and the environment as factors that can impact health and disease. And we've been trying to think about these questions in so many different contexts. But the focus of my talk today is going to be specifically on immune response and disease susceptibility. And if we think about the impact of genetics on disease in particular, we like to think about it from a quantitative genetic perspective, which means that genetics will have a role, environment will have a role, and then interaction between the two also can be extremely important in this context. One of the most exciting developments in the field was multi-omics. It's the idea of integrating different types of omics data to bridge genetic variation and variation that we see in health and disease. And this is extremely important, even as we zoom in into cells and how cells function. If we think about the various inputs that our cells and organs and systems are exposed to, it's exactly the same framework. It's genetic and environmental impacts that will define how these cells would respond to different types of stimuli or just to normal biological processes like aging, for example. So now from an evolutionary perspective, and throughout the history of humans have undergone very significant lifestyle transitions in their lifestyles. The most significant ones was the Neolithic Revolution, where humans have experienced this marked shift from hunter-gathering to settled agriculture. And this happened around 9,000 years ago in the Fertile Crescent. Now, more recently, urbanization has contributed to the rise of complex diseases. So as of 2010, the number of people who live in the cities, urban lifestyles, surpassed the number of humans who live in traditional lifestyles, like urban or nomadic lifestyles. And this transition coincides with this massive increase that we've seen in the incidence of complex diseases. So now our genes are not going to change that quickly. You need many, many generations for genetic variation to change at significant differences in the frequencies. So it's extremely important that we think about changes that we have in the environment as being extremely important on top of genetic variation that act on these diseases. And I think we can all agree that genetics is extremely important. There have been hundreds and thousands of studies that have been published that made a very specific link between both rare genetic variation and common genetic variation, whether we are talking about rare diseases or common diseases. And we've seen this massive explosion in the number of genetic variants that have been associated with complex diseases, specifically with GWAS studies. Now, I'm just showing you the map of the human genome. These are the human chromosomes, and these are the number of GWAS discoveries that have been reported in 2009. In 2010, we started seeing more GWAS studies that have been published. And now we see basically hundreds of thousands of genetic variants scattered across the human genome that have been associated with complex diseases, as well as just quantitative traits. And many of these quantitative traits are the underlying types of variation that can actually drive disease. Now, again, we can all appreciate that genetics is important, but we like to think about our genetics really defining our baseline risk. And the point I want to make today is really about the importance of the environment acting on genetic variation. So if you think about different human populations being exposed to different types of pathogens, including microbiome as well, different populations can live in extremely very different geographies or climates, having been exposed to different types of food and drugs. And so all of these environmental exposures, again, acting on our genomes is really what defines health and disease. And we've been trying to think about these questions for over a decade now. Since I did my graduate work, I started the first study to investigate these questions, and my focus was the Moroccan population. And Moroccan population is very interesting when it comes to its genetic makeup, because it's really a highly admixed population. We have the Amazigh, sometimes we refer to them as the Berbers, like the indigenous people of North Africa. Then you have the Arabs, and then you have a sub-Saharan component. You have even contributions from back to Africa migration from Europe. So it's an amazing system when we think about genetic variation. But also, interestingly, the populations of Morocco experience a wide range of lifestyles. And my focus initially was to specifically compare the profiles of people who live urban lifestyles versus people who live traditional lifestyles. And we've conducted a couple of studies. One of them looked at nomads as well as people who live in the Atlas Mountains. And a second study where we focused completely on just comparing the profiles of individuals who live very traditional lifestyles and people who live urbanized lifestyle. So this was already published over a decade ago, and we really looked at, again, focused on these two specific lifestyles. And what's really important to remember is that with humans, it's a little bit complicated to actually control for confounding between genetic variation and environmental factors. So we have to really use statistical genetic methods to be able to account for effects that can be attributed to genetic variation versus environmental factors. And we looked specifically at gene expression of like peripheral blood. Peripheral blood, where you have circulating immune cells, are a very interesting model to actually study these gene environmental effects on gene expression. I won't have time to go really into the details of the study, but I really want to highlight the fact that what we really discovered pinpoints specifically to the very strong impact of lifestyle on susceptibility to disease. And I should mention, in this study, we focused on healthy individuals and not individuals who actually have disease. And we confirmed this with using clinical chemistry tests in the lab. So this is extremely important. Again, you have populations where we control for genetic variation. The main difference is mainly in their lifestyle. Of course, geography is confounded with lifestyle in this context, but we have very strong evidence that really it's lifestyle that impacts disease susceptibility in the context of at least biomarkers of disease that are expressed in blood. We're expanding this project. So very recently, we initiated an expansion of this study. This is a study that is funded by the Sean Zuckerberg Initiative, and we are going back to the same population where we sampled 50 individuals who live urban lifestyle and 50 individuals who live traditional rural lifestyles. And we are doing this now at single cell resolution, where we are actually extracting or collecting blood samples and isolating PBMCs, and then we can look at different differences between these two groups, both looking at epigenetic and genetic factors. And we are also doing ex vivo experiments in the lab where we can expose PBMCs of these individuals in the lab and see how they respond to these mimicking basically bacterial infection using LPS. And again, the idea is to see how the immune response of these individuals, while we control for all the variables, can be different, which could in subsequently drive susceptibility to disease. And as Andrea mentioned earlier, we're also expanding our work to Mongolia. So more recently, last year actually, we started doing field work in Mongolia. And Mongolia is very interesting when it comes to the lifestyle of the population, because you have almost half of the population live traditional nomadic lifestyle, while about a third to a half of the population live mostly in the capital city of Mongolia, Ulaanbaatar. And we know from epidemiological studies that there are differences in terms of the prevalence of diseases. So here you are talking about a population that is making a transition from traditional nomadic lifestyle to urbanized lifestyle with all the variables that we can think about in terms of diet, for example, or exposure to stress and other variables. These are just some pictures from the field work that we conducted last year. And it was a really interesting time that we spent with the nomads to really experience how unique their lifestyle is. And I want to pinpoint specifically diet, because diet is an extremely important factor that I would like to go back to and discuss if we have time later. All right, now going back to the Middle East. So the Middle East is one of the populations that experienced very, very significant transitions in lifestyle. So here I'm showing you pictures from 4 cities in the Middle East from the 50s and 60s, and here where we are now. So here you are looking at Abu Dhabi, Doha, Riyadh, and Dubai. And again, we know that the incidence of cardiometabolic disease is almost epidemic in these populations. And again, genetics of the population is not going to change that fast. You need many, many generations for the genetic, I'm talking about DNA variation specifically, for the frequency of mutations to change. Epigenetic can change. The epigenome of individuals, of course, can change as a response to the environment. But the most important factor here is purely environmental related to lifestyle. So to be able to study these factors in the Middle Eastern population, we set up a study called the UAE Future Study. It's the first prospective cohort study here in the Middle East. We started this study almost 10 years ago. It's having these prospective studies is extremely important because it will allow us to collect baseline data, and then our study participants agreed to be followed up with the idea of collecting information every 4 to 5 years from this cohort. So we're now at that stage where we're about to actually complete recruitment for the baseline cohort, and we will soon start the follow-up of the study. The study is really targeted towards sampling the general population, adults, anyone who's Emirati, like a local UAE citizen, and 18 years of age or older, they can enroll in this study. And again, we do a series of tests, and this including collecting data about health, we have a lifestyle questionnaire, demographic data, family and personal health history, and then we collect blood samples for clinical measurements. We do blood profiling. We also collected urine samples. I should mention the size of the cohort is about 14 to 15,000 individuals. And now we are still in the process of finishing the recruitment of the baseline cohort. And we collected urine samples from about 5,000 to 6,000 individuals, and mouthwash samples as well, specifically for all our oral microbiome studies. And our plan for the follow-up phase is to do both a passive follow-up, where we can actually link some of the cardiometabolic disease outcomes to the baseline data that we already collected. And then the active follow-up involves calling some of the study participants back to the clinic and then collecting more samples and expanding some of the phenotyping that we do to things like ECG, OCT, and brain MRI scans as well. So now I want to switch again. I want to really take you to another continent. Now we're going to Africa, just because I want to make the important link about the importance of diet. So my lab has been interested in studying immune response specifically, and we decided to do this in the context of malaria. Malaria is a global health public problem. It's not an epidemic. Every year, at least 600,000 people die of malaria. And the majority of these are children under the age of 5. And I'm not even going to talk about the loss in productivity worldwide, but again, this is a big problem, mostly in sub-Saharan Africa, as well as Southeast Asia and Latin America as well. So what we decided to do is to use a quantitative population genetic approach. The idea is to really dig in and zoom in and use multi-omics. And here we have a system. If we are looking at blood stage malaria from a blood sample, where you have the immune system that tries to control proliferation of the parasite, you have the parasite that tries to proliferate, and all of this can be modulated by these molecular mechanisms that involve both like metabolites, lipids, transcription, translation. All these processes basically are taking place within the blood sample or the blood tissue. And what really triggered us is this, what we know from epidemiological studies as to the difference that we see between populations. We know, for example, that in Africa, some ethnic groups are more resistant or susceptible to malaria infection. And that was always a very intriguing question. Why? Is it genetic or is it the environment? So we decided to set up a study where we've been recruiting children specifically from 3 ethnic groups, the Gwan, the Musi, and the Fulani. And the design of the study is really based on prospective sampling, which is a very, very powerful study design. We do our work in Burkina Faso, and in Burkina Faso, malaria is seasonal, meaning that you have a dry season with very, very low transmission of malaria. And then when the rainy season kicks in, that's when transmission starts taking place. This gave us really a very interesting model in which we can actually sample children before they get infected around the end of the dry season, and then we follow them over time. We collect blood samples from these children. on a weekly or a bi-weekly basis. And then we do the blood test, we use microscopy. And if we detect the parasite in their blood, we call them the asymptomatic parastemia group. And then we keep following them till they have typical malaria symptoms, and we sample them again. Only then they are given treatment, and we sample them again 3 weeks after treatment. So we can study a lot of phenotypes related to how these children respond to malaria infection based on this design. These are just some pictures from the field. So we set up a lot of connections with the local population. We have very successful collaboration with the local team there in Burkina Faso, because we also do community outreach. We engage with the population. We also help capacity building in Burkina Faso. You can imagine that these are very, very challenging conditions to do molecular or genetic work. But we did actually manage to set up or have the setup there to be able to do molecular work. And as you can imagine, this field work is extremely challenging. Sometimes we get lost in the middle of the jungle. This is a day where study participants have been waiting for us, and we couldn't make it to the village. But again, this is what happens when we try to do things in the field. A lot of unexpected events can take place. We also had to adjust to the facilities available to us to do sampling. This is an instance where we had access to a school, where we actually use the school to be able to recruit the children and phenotyping, as well as collection of blood samples. I just want to highlight that the conditions in which these populations live can be extremely challenging for them. Every time it rains, this is a very prone environment for malaria transmission. And I should mention that initially we recruited 150 children to our study, estimating that potentially around 100 of them would be infected during the rainy season. What happens is that all of them got infected. 150 children got infected with malaria. And this is just to give you an idea about the level of exposure to malaria in these populations. And I want to mention specifically the Fulani ethnic group. They are very interesting when it comes to malaria. They have always been reported to be less susceptible to malaria, and we've never really understood why. There are some studies that have been published that showed some very interesting characteristics when it comes to their immune response that they have, including lower Treg cell activity, as well as higher level of interleukin-4, interferon gamma, as well as high levels of antibodies against Plasmodium. They've also been reported to have lower parasite gametocyte counts. So during the blood stage of malaria, the very last stage that the parasite undergoes is gametocytogenesis, where the parasite produces gametes, and those gametes are taken up by the mosquito, and that's what sustains transmission. And we never really understood why this ethnic group tend to have these low levels of gametes, of parasite gametes in their blood. So because we work with children and relatively small volumes of blood, we developed a lot of methods to be able to maximize how much molecular data we can obtain from these samples. And we did manage to really kind of make the most out of these small volumes of blood samples, so that we can do DNA work, RNA work. We extract serum as well as whole blood and PBMCs. These samples allow us to really do almost a full range of genomic applications. From the infected children, we can also collect molecular data from the parasite, because the parasite, as I mentioned, it's in blood. So we can actually look at the genome and the transcriptome of these parasites. So the first study that we published was a few years ago. And in this study, I won't have time to go into all the findings in the study, but I want to highlight something very specific. In the Guan ethnic group, one of the 3 ethnic groups that we studied, what we've shown is that when the children are infected, they trigger steroidogenesis. So steroidogenesis is an indigenous signaling pathway in T cells of these children that actually produce a number of steroid molecules. We're not talking about drugs here. We're talking about steroid molecules that are synthesized from cholesterol all the way to sex hormones, and the same pathway produces these steroid molecules. These steroid molecules can be very potent because they have very strong anti-inflammatory properties. And what we've noticed is that in the Guan ethnic group, these children, upon infection, they trigger this steroidogenesis. And this would have very significant implication on their immune response, because if these steroid molecules are produced, that literally dampens their immune response, which gives the parasite the opportunity to proliferate. And that's the association that we detected in the Guan ethnic group. But on the other hand, when we looked at the other ethnic group, the Fulani ethnic group, the one that is less susceptible to malaria, we've seen the opposite effect, as shown here in this volcano plot. So these steroid molecules are highlighted in the red. So what you see in the Fulani ethnic group, it's either no change or actually these steroid molecules going down, which again, we don't understand the reason why, but it's an observation that we at least validated using an independent data set. Now, one of the other things we wanted to look at more carefully is the impact of diet. And to look at the impact of diet, we focused on the Fulani and the Mossi ethnic group. Why? Because they are actually sympatric. They live in the same environment, the same neighboring villages. So environmental exposures to malaria and to environmental and geographical factors are the same. The only difference here is ethnicity. And the single most important factor that actually differentiates the two ethnic groups, apart from genetic differences, is diet. Fulani are nomads and they consume a lot of dairy-based diets. On the other hand, the Mossi, they consume mostly plant-based diets and almost no dairy-based products. So for this study, we recruited 53 children from the Fulani and 45 from the Mossi. It's the same study design. It's a fully matched study design, meaning we collected blood samples from these children before infection and then during infection. And we've done everything the same way so that we don't suffer from any confounding or like batch effects. Everything was randomized in this experiment. So to be able to really look at lipids specifically, because lipids, it's really the most revealing, like omics type of data that can tell you about diet, we used high resolution lipidomic analysis. And it's very similar to mass spec, but it's actually high resolution because the method that we used can actually tell you exactly the fatty acid composition of most of these lipid molecules. And there are over 900 complex lipids that we identified in this experiment. And the question here is, okay, what has more impact on the lipidome of these children? Is it ethnicity or is it infection? Again, because we have children who have been sampled before infection and during infection from both ethnic groups. This is principal component analysis of the lipidome. And what you see in the first panel to the left, labeled by ethnicity and by infection status, what you see here is that the clustering of the lipidome is based on ethnicity and not based on infection, which means that the effect here is stronger. The effect of ethnicity is stronger than the impact of infection on the lipidome. And we see when we split the two data sets based on infection and non-infection, you see exactly the same pattern. You see this difference that you see between the Mossi and the Fulani. And when we dig deeper to really identify what are the lipid molecules that are different between the two ethnic groups, here I'm showing you a heat map in the middle that shows the most significant lipids that are actually different between the Mossi and the Fulani. Most of them are very interesting, but what really triggered our attention is the fatty acid that is associated with these lipid molecules, which is linoleic acid. Pentadenoic acid, sorry. It's I'm just going to highlight one of them, the second one from the top. It's called LPC15. This is lysophosphatidylcholine, because this molecule has already been reported to actually block gametocytogenesis. This was reported in a Cell paper from 2017. And again, infected or not infected, the levels of these molecules is higher in the Fulani ethnic group. Now, what's even more interesting is when we look specifically at the LPC15 and pentadenoic acid, which is the fatty acid associated with this molecule, it comes from milk fat, which makes perfect sense. It's exactly what we know about the diet of this ethnic group, the Fulani. So the fact that the Fulani consume a lot of milk, the concentration of these molecules that contain these lipid molecules with pentadenoic acid, C15-0, have higher levels in the serum of these children. Which, if you remember what I mentioned earlier about the report that gametocytemia tends to be lower in the Fulani, what we decided to do is to go back to the lab and try to see if we can actually functionally validate the impact of this very specific lipid on gametocytogenesis in the lab. To do this, we do a parasite culture. And again, I won't have time to go into the details, but bottom line, we show exactly that the impact of this LPC15 is blocking gametocyte production in the serum of these children, which we can, as I mentioned earlier, link to the fact that the ethnic group has lower gametocytemia in their blood. So I hope I convinced you that both genetics and environment are extremely important. We're really excited about diet now because now we are at that stage where we have the methods where we can identify very specific types of molecules that can come purely from diet, which means that the impact of genetic here is going to be very low or minor, except if metabolism is involved. We have to remember that the microbiome and our genes can be important in how our system, our body, can actually process some of these lipid species. I'm running a little bit out of time, so I'm just going to mention one more study very quickly, where we compared the same two ethnic group, but now we're actually using single-cell genomics specifically, as opposed to bulk RNA-seq. And what I want to mention is when we do more or less the same study, where we compare the Musi and the Fulani, this is UMAP, where we have UMAP of about over 70,000 single cells, PBMCs of both ethnic groups, and we did compare the PBMCs of these children before infection and during infection. And again, what was striking to us is the impact of ethnicity relative to the impact of malaria infection. This is a PCA that just shows what we call the pseudo-bulk data, but it's basically from the single cell data, but it's pseudo-bulk, which means we aggregate all the gene expression data across the different cell types. And what we see here is, again, if you look at the colors, the blue and the red, that's differentiating the two ethnicities. And the impact of infection, as you can see from the light and the dark color, is minor relative to the impact of ethnicity. And we see this when we look at specific cell types, like here I'm showing you CD14 and CD16, we see exactly the same story. But we almost see this across all the cell types in these children. This is just a heat map to give you an idea about the significance of differences in gene expression between these two ethnic groups in the infected state. We expanded data analysis to look at the impact of genetic variation. I want to highlight specifically the fact that we didn't have a lot of power to actually look for what we call the SNP by ethnicity interactions. But I'm highlighting two genes here where these are two genes that are actually very important in the context of malaria pathobiology. They've already been associated with malaria. And what we discovered is that the individuals of the children respond in terms of their expression in a different way depending on their genotype. So if you look at the individuals, at the children that have the minor allele, referred to here as 1, you can see that the difference in expression is seen in this group between the two ethnic groups, between the Musi and the Fulani. While the individuals who have the minor homozygote, the major reference homozygote, we don't see any difference between the two ethnic groups. This is very intriguing because this is again an example of how ethnicity can interact with our genetic variation to drive gene expression of some of these genes. So finally, I would like to acknowledge our study participants in Burkina Faso, in Morocco, in Mongolia, and the UAE, and as well as my lab team members who did all the work, starting in the field all the way to experiments in the lab and computational analysis as well.
Thank you so much. What a wonderful presentation with many countries. We actually crossed many borders, many ethnicities, many genotypes. Thank you so much. Let me ask you the first question. How could we, knowing that ethnicities are now crossing borders and we are living in same environments, think about Singapore, Malay, Indians, Chinese, Caucasians, how do we optimize our lifestyle based on our genome to have a maximum outcome, which might be health or should be health?
Yes. Yes, that's I mean, that's a very good point in terms of even thinking about personalized medicine, because we can go all the way from populations all the way to the person itself. So the idea of personalized genomic or personalized medicine doesn't even take into account ethnicity, because we're looking at individual genomes, individual genetic variation, individual lifestyle. What's interesting about ethnicities is these common things that sometimes cannot be captured at the individual level. To give an example, in our case, diet was the single most important factor that really distinguished the Mossi and the Fulani. So we're using that to our advantage to make discoveries. Once we discover things that are unique about certain ethnic groups, and here again, I have to emphasize that ethnicity is what we think about ethnicity as a self-reported information, which can complicate things, because we can think about genetic ancestry, but here we're talking about ethnicity as, you can sometimes have ethnicity being driven by diet and genetics. You can have other populations where it's the opposite, where you have the same lifestyle, the same diet, but differences could be genetics. So I think having like group specific type of interventions is something where probably the field is beyond that, because now we're at that stage where we can do personalized interventions, for example. But at least for the sake of discovery, if we take advantage of these differences that we see between different ethnicities and different populations, that can really help us pinpoint what's important.
And how would then the most successful study design look like? How would you design a study to optimize what we do? And it's not the WHO guideline, which is just on population level and very crude, I would say. It's good, but everybody should do it. But how do we then optimize our interaction between where we live and what kind of book, the genome we have?
Good point. I think, at least in the field of genetics, the idea of doing a mixture mapping was the first actually instance where we saw the power of doing this as mixture mapping. Like if you look at African Americans, for example, they range between Europeans and Africans, and individuals can fall anywhere between the two extremes. South Americans, it's the same thing, if not even more complex, because now you're adding an indigenous component to the population. And then you have all these debates about how do we even define ethnicity or genetic ancestry. I think we are beyond, at least in the field of genetics, beyond this categorization, because we know that most individuals are actually admixed when it comes to genetics. So genetic variation is more of a continuum, more than actually subjective ways of grouping people. So what I would say in terms of the design of the studies, at least information should be collected. I think as much information as possible should be collected in these cohort studies. We're often lacking information related to ethnicity, broadly speaking. Genetics, inferences can of course be made from genetic data if genetic data is available. But I think in terms of intervention specifically, and I'm gonna use the malaria study that we've done as a good example, because the fatty acid that we discovered as being correlated with resistance to malaria is something that can actually lead to a very specific intervention. Now, some suggested that we should do a clinical trial and change the diet of the Mossi, but it doesn't have to be that extreme. It could be even a supplement. The beauty of some of these lipid molecules, they are safe because you find them in regular foods. So an intervention could be based, for example, not at the individual level, it could be at the population level, in terms of like just giving these fatty acids as supplements and then looking at the impact at the epidemiological level. Now, at the individual level, I think what we can do, and there are studies that have shown this, is to look at why some individuals respond to some interventions and others don't. And we know this. It can be driven sometimes by genetics, it could be driven by the microbiome. But the hope is that we should really emphasize the importance of lifestyle in this, because genetic information or genetic variants often tend to have a minor effect, as opposed to interventions related to lifestyle or diet or physical activity or other. And it's not that complicated as opposed to like genetic like doing something related to genetics. I think the talk earlier about CRISPR is very exciting in terms of understanding mechanisms of disease, but we're not there yet at the population level of expanding these type of maybe rare diseases. Yes, I mean there are studies that have demonstrated that at the individual level we can use these genetic variants, but they tend to be extremely rare, not population specific. So in that context, ethnicity or genetic ancestry is not important. It's a single mutation that is important. I should mention here in the UAE, for example, they started a premarital screening that uses information from the Emirati Genome Project. Now, what's really interesting is that even susceptibility to certain cardiometabolic diseases can be driven by alleles that are more frequent or less in different populations. And that's where things can get complicated, because you can have an allele that confers resistance, another allele that confers susceptibility at the individual level, or the product of both. So, yeah.
Yeah, interesting. You touched on the genetic variation and the change, and that takes a long, long time until we have other genetic variation. Could we think about scenarios where that transition is not as long, but could be, by forward with, for example, a huge stressor, could be much, much more intense? And do you have examples, any out of the wildlife or with humans?
You mean stressors in terms of their impact?
Diseases, for example. Yes. So how do we actually, how you said it's taking a long, long, long time until we really adapt the genetic variation. And I think what you're talking about is selection. How much selection is there actually while looking at genetic variation? And don't we, because of good healthcare, actually reducing the genetic variation?
Well, yeah, I mean, it's at least in humans, the cases of like positive selection, they are there. The lactase story is a good one. There are examples from the immune system where selection can be extremely strong, but it's really minor to what we initially thought in the field of science. So most of the variation in the human genome is actually driven by negative selection. The thing with modern medicine and the fact that even the strength of selection because of modern medicine is lower, we increase the number of these deleterious mutations. And now, even if you think about the Middle East and other countries such as Pakistan and others, where levels of consanguinity are high, at the individual level, that's where we increase, for example, homozygosity, which can increase again prevalence of some of these conditions. But again, it's a very different story whether we're talking about rare diseases or complex diseases. Definitely complex diseases can, if we understand the genetic baseline risk, and if we can at least stratify individuals based on their baseline risk, then some individuals can be prioritized for certain preventions or interventions other than others. I think this is the most promising use of genetic data at this stage. But in terms of the magnitude of the effects, definitely lifestyle has the strongest impact.
I think that's absolutely clear. Thank you so much. Before handing over to Wahan, because I think that there are lots of questions and he is already scrolling through, I have one more question. I really loved your note that you do capacity building wherever you are. I think this is so important. So thank you so much for sharing it. To really also democratize research. I always think we are talking about democratizing health, etc., and healthcare, but I think really democratizing also the knowledge we have. Can you share us the most successful capacity building project you ever did during your research?
I think that that's definitely something I'm very passionate about. I started doing that in Morocco during my PhD work. I was an exam product myself of that process. And then I think what we did in Africa in Burkina Faso is really was extremely important because yes, we do research, we look at genes and genomes, but we're working on individuals and populations who are suffering big time from these conditions. And sometimes the gap between research as we do it in developed countries can be huge. And we should not lose sight of what could be extremely important from these populations. So, for example, things as simple as PCR or genotyping can really help in terms of both detection and surveillance programs. So the setup that we have in Burkina Faso is really giving them the capacity to be able not just to learn what we do as part of the project, but to expand those resources to what they do. And I should mention that we will actually start similar program in Mongolia. For example, next year, we will run a workshop on microbiome. And this has huge implications for them because looking at the microbiome and looking at antibiotic resistant is something of utmost importance to them. We have to be very careful how to transfer knowledge in a way that they are actually able to do it 100% there, as opposed to if we teach people how to do whole genome sequencing, that's useless. Let's say in Burkina Faso, because they don't have that type of data. So we're trying to tailor what we do to exactly what are their needs. And when we do this, we really push to have students and local researchers involved in doing this. The Mongolia project was actually jump-started by 2 Mongolian students in my lab, and they have plans to actually go back to Mongolia and set up this type of research that is adequate to the questions and resources, importantly, resources that they have there. But I think as a community worldwide, we can contribute in so many different meaningful ways.
Yeah, love it. Thank you so much for sharing that. Well, I think lots of questions. Over to you.
Thank you. And thank you, Professor Youssef, for your fantastic speech. And we have a lot of questions from the panels. So the first question is what challenge do you actually face when you're working with this genetically diverse populations like those in Africa or UAE?
So we actually use genetics to our advantage. Yes, you have a lot of challenges in terms of data analysis. In terms of data analysis, we have to be careful. How do we distinguish genetic factors from environmental factors? So the design can help us make sure that we can disentangle this genetic and environmental effect. But we actually gain more power by working on diverse populations. So most of the discoveries that we made really stem from the fact that we have diversity at the genetic level between individuals. Without that genetic variation, we won't be able to actually detect or at least discover some of the signals that we got from the data. So yes, it's challenging, but if you use the right study design, if you use the right statistical genetic methods to be able to account and disentangle environmental and genetic effect, I think it's way more powerful than what could cause a problem for statistical inferences.
Thank you for your answer. And the second question will be about the personalized medicine. So how could your research potentially inform public health interventions or personalized medicine strategies?
So I'm going to use the malaria as a good example. Everything that we've done was really a complete zoom out. The reason why we looked at lipidomics is that we didn't want to miss anything. It's not even hypothesis driven in terms of what lipid we're looking at. Then when we made the discovery of this fatty acid, this pentadenoic acid, or we have another study that I'm not even discussing today, where we discovered another lipid species which can actually help the parasite proliferate. But let's just limit I'm going to limit my answer to the acid, the lipid that blocks the parasite. This is a lipid that can be bought. It's quite cheap. And this can be now implemented as an intervention studies. We're actually negotiating now with the authorities in Burkina Faso about the possibility of running like a clinical trial, which is safe because it's not a drug, So we can be easily, if we can do like a randomized controlled trial, this will tell us if we are actually reducing transmission of malaria or not. It's a very simple question, a safe compound. We don't even need people to drink milk. They can use it as a supplement. And then if it's a controlled trial, we should be able to at least see if we're reducing transmission in the population or not. That's one example. And there are many, many other examples that stem from our research.
Thank you for your answer. So we have a futuristic question. So what are some of the emerging technologies or analytic methods you're excited to integrate into your future studies?
Good point. I think apart from lipidomics, which we're really excited about, because lipids, I always thought about lipids as they only make membrane, but they're actually very important molecules. They can be very potent when it comes to their regulatory potential. So lipidomics is a very, very exciting field, and I think we're going to see a lot in this field. Of course, proteomics now is taking off as a field. Microbiome studies, we're at that stage now where sequencing cost is relatively low. So we're now seeing this transition from doing 16S to doing metagenomics. So the idea of doing metagenomics, the idea of actually sequencing the full genome of gut microbiome, for example, is very exciting, because that's how you start to really making a link between genomes, diet, microbes, and physiology and disease. So I would say proteomics, lipidomics, and microbiome are the three most exciting fields right now in terms of at least cost and the ability to generate high quality data.
Thank you for your answer. So the next question is that you have mentioned in the talks that their populations often exhibit chronic immune activation, which is potentially due to the continuous exposure to the external pathogens and lifestyle related factors such as diet and physical activities. So how in your study you are actually controlling these confounding factors?
Which one do you say? Which one?
The confounding factors. So it's basically the exposure to the external pathogens and lifestyle-related factors.
Yes. So for exposure, because the population we are studying are in the same geographic location, they actually have the same levels of exposure. And because we are sampling children before infection and during infection in the same geographic location, we are controlling for exposure. We also control for the potential differences in the strain of the parasite, because that's another confounding variable. How do we do that? By doing the genome sequencing of the parasite from whole blood. So we don't have any differences in terms of strains, for example, different individuals or different populations exposed to different strains. Everything else, we try to really control for it statistically speaking. If there is a proxy of the environmental exposure that we can actually measure in blood, we can use it as a covariate in our statistical model. To give a very specific example, we use a total white blood count as a variable in our model. And that, in principle, it should control for differences that could be attributed to maybe other co-infections. Maybe some children might have actually a viral infection, or they might have a bacterial infection. So it's not perfect. We are talking about samples that are collected in vivo in Africa. So you will definitely have some variation due to factors. The best we can do, we minimize it through our statistical analysis and also avoiding batch effects in terms of being able to process all the data at the same time instead of processing it at different times.
Thank you for your answer.
Just one thing I should mention. I really want to emphasize this prospective matched study design, because it's the most powerful. If you have the same individuals sampled before and after, or at 2 different time points, you're actually controlling for a lot of variables just by doing that. Because it's the same individual, same age, same sex, same diet could be different, but we actually control for that because we can collect questionnaire data, but matched or paired study designs are the best to make statistical inferences.
I see. Thank you for your answer. So we might not have enough time. So the one last question is that, have you ever found like evidence of inherited COVID-19 immunity in your immune cell atlas research?
Our short answer is yes. There are some genetic variants that have, I'm not talking about common genetic variants, because we all have those common genetic variants, but their impact on COVID-19 susceptibility is very, very small. But there are instances of rare genetic variants that have been discovered that can explain a number of COVID-19 cases. I'm talking about severe COVID-19 cases. So there was a science paper that was published after the GWAS study of COVID, and in that science paper, they focused specifically on these rare variants of like very significant impact. And we know just from genome sequencing, and I'm talking here about some of our data sets, we identified few individuals that are carrying the allele that increases by 5 times the risk to COVID-19. And again, these are genes that are very important in immune response, and they have a very significant impact. But those are rare in the population. The majority of COVID-19 susceptibility is driven by hundreds, if not thousands, of SNPs, but each one is contributing minor effect.
Thank you for your question. So we might have enough time. Is there anything for Andrea?
One short question if you have one more. Burning, short answer. Do you have one?
Can the C15 supplements help reduce malaria?
I truly believe it has a lot of potential. I really believe, because it's a safe molecule and it blocks transmission. And that's what we need. We just need to block transmission. If we do that in a safe way, I think there is a lot of potential. But again, I don't want to make it sound simple. Malaria has been here. I don't know if you guys know, but malaria arguably is the #1 killer of humans. Arguably, it killed billions of people throughout the human history. So yes, it's probably here to stay, but what we need to do is just to reduce these rates, because still over 600,000 individuals die every year. And we need like different strategies. We need maybe supplements, maybe there is hope that the vaccine that is currently being tested in Africa will work. But the parasite has shown us that it's so clever in how it evolves and adjusts to all sorts of interventions. So I think we need a collective effort that tackles the problem from different angles. But definitely supplementing the diet, for example, with the C15, could potentially reduce transmission. And that's all we need. Just reducing transmission will save a lot of lives.
I think that's a wonderful message you have. I think if there are foundation listeners, if there are VCs listening, you want to fund these type of research, please be in contact with Professor Idaghdour. Thank you so much, Youssef. It was a wonderful presentation about the topic we don't think so much about, because so much of our research is not being democratized yet. And I think we should really focus also on other regions and countries where we look at aging processes to really understand not only human behavior, but also the genetic component and the environment interaction. So thank you so much for your scientific work and the capacity building work. You as listeners, hopefully you also enjoyed this presentation as I did. So use the chat function and choose the panelists and all attendees option to leave your comments, your feedback, who you would like to have in the show. We are very welcoming your feedback, because that's very, very important for us. The next Healthy Longevity webinar is with a dear colleague of mine on the 3rd of July. This is Dr. Nicole Sirotin of the Institute for Healthier Living. Also, yes, we are staying in Abu Dhabi. As always, we are closing this session with a final video, and that video is sharing a couple of very simple tips. So you might want to introduce it. Take care.
Behavior is the sum of everything we do, and what we do is influenced by who we are, the people around us, and by our environment. In fact, our behavior consists of all the choices we make throughout the day, and of course, our habits too. We know that some of our behavior increases the risk of getting ill, while others help us to stay in good health. And it's those behaviors we want to stimulate, right? Changing our behavior towards a healthier lifestyle might seem hard, but it's definitely something everybody can do. To change our behavior, we need the ability to do so, the right opportunities, and we need to keep being motivated about it. Scientists have come up with a few handy tips to help us facilitate the process of behavior change. Try and see what works best for you. 1. Set goals, making them specific and realistic, like a 20-minute daily walk, rather than just aiming to walk every day. Step by step. 2. Reflect. More veggies or be more active? Reflection will give you a clearer picture of what to change and how to get there. 3. Make it easier. Simple things like bringing your sports bag to work or having smaller portions can make a big difference. 4. Get support. Tell family and friends and let the support roll in. It might even make them start their own behavior change. 5. Get active. Do something you enjoy. Moving should be fun. Woohoo! 6. Plan. Planning your meals and exercises will reduce stress and make the transition to your new lifestyle smoother. 7. Reward yourself. Set a milestone, and when you reach it, allow yourself a treat or a fun activity. 8. Monitor your progress to encourage yourself, either in a diary or on social media. Changing behavior is rewarding in so many ways. Instant satisfaction, liveliness, an improved social life, and you will also be rewarded later on with better health. Don't postpone. Start changing today.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.